Hi, everybody. Welcome to the BrainSpace Initiative. It's our pleasure to introduce Dr. Yu Zhang, who's an assistant professor in psychiatry and behavioral sciences at Stanford. And he actually has an engineering background because previously he was a professor of engineering, bioengineering, and electrical and computer engineering at Lehigh University. He's been doing a lot of work in data-driven neural pattern decoding and biomarker discovery, as well as working with multimodal neuroimaging. So it's very near and dear to my heart. So I'm looking forward to seeing what you have to share with us today. So I will let you take it from here, and thanks so much for joining us.
Thank you so much, Vince, for the nice introduction and for having me here. And it's a great opportunity to kind of share with everyone some of our recent research updates. So my talk today is about unlocking precision mental health with data-driven neuroimaging biomarkers. So here's a quick overview of our research focus. So by leveraging advanced neuroimaging and cutting-edge AI technicals, my research primarily focuses on developing clinically translatable biomarkers for precision diagnosis, prognosis, and personalized intervention of mental disorders. So in my presentation today, I'm going to primarily cover three different aspects relevant to our recent research progress. So including treatment predictive biomarker quantification for personalized medicine of mood and stress-related disorders, and also establishing brain behavior mapping of psychiatric heterogeneity in neurodegenerative and neurodevelopmental disorders. And also quantify brain signatures for substance use disorder, in particular cocaine use disorder phenotyping. So mental disorders are among the most prevalent conditions across our lifespan and place a significant burden on individuals, healthcare systems, and the society. Traditionally, diagnosis relies on clinical observations and behavioral symptoms. However, such subjective framework has caused a substantial heterogeneity and overlooks frequent commonalities across disorders. So those limitations hinder both our understanding of underlying mechanisms and the development of targeted treatment. And so currently, there's still a lack of objective biomarkers and personalized treatment guidelines for mental disorders. And with advances in artificial intelligence and neuroimaging, so there's a growing interest in using machine learning or computational modeling approaches to enhance precision medicine. So this type of approach, hold great promise to shift the conventional one-size-fits-all models toward more personalized interventions that can match treatments to individual patients and ultimately maximizing clinical benefits. So first, I would like to briefly talk about the several of our studies aimed at developing neurobiomarkers of antidepressant response using data from a large randomized clinical trial. So major depressive disorder, or MDD, is a leading cause of disability worldwide, with a lifetime prevalence exceeding 20% and affecting over 200 million people globally. So although antidepressants are widely prescribed as a first-line treatment, their efficacy is modest, often only slightly better than placebo for a large number of patients. So this limited efficacy is largely due to neurobiological heterogeneity within the clinical diagnosis. So identifying more objective biomarkers to guide a treatment selection is really important and critical for improving treatment outcomes. So to address this challenging, we use the data from the EMBARK trial, which was designed to identify moderators and biosignatures of antidepressant response in major depression. So over 280 patients were randomized to receive either antidepressant sertraline or placebo for 8 weeks. Baseline assessments include both FMI and EEG, and treatment outcomes were measured by pre and post-treatment HAM-D scores, which is a golden standard to assess treatment effects in major depression. So here, both resting FMI and EEG capture spontaneous brain activity, while task FMI provides a window into the functional process. So in this case, the emotional conflict regression, which is a cognitive emotional function often disrupted in depression. This task involves regulating conflicting emotion stimuli and requiring adaptive engagement of a cognitive control process. So previous studies have shown that depressed patients usually struggle with regulating conflict-related brain activity. So here, our first step was to examine whether brain responses during this conflict regulation could review phenotypes that differentially predict a treatment response. So we examined the pre-treatment fMRI activation as a predictor of antidepressant outcome versus a placebo outcome. So using a whole brain voxel-wise linear mixed effect model analysis. So here we identified a significant treatment moderation effects in several key brain regions, primarily including bilateral DLPFC, frontotemporal cortex, DACC, and insula, as well as hippocampus, visual cortex, and cerebellum. So which are all part of canonic conflict response networks. So to better interpret these effects, we split the participants based on their level of regression-related activation using a median split. So we found that lower activation during emotional conflict regression, for example, in the DACC and frontotemporal cortex, were associated with greater treatment or clinical benefits from antidepressant treatment. So this actually suggests that a blunted engagement of conflict regression circuits may be useful biomarker for predicting treatment response. And then we conducted a machine learning-based multivariate analysis to predict individual level treatment response. So we built a multi-atlas sparse regression model to predict symptoms change at the individual level. So using a 10-fold cross-validation, the model trained on the sertraline arm significantly predicted a sertraline treatment response. However, it didn't generalize to the placebo arm, as you can see here. So this actually indicates that the model captures a sertraline-specific signal related to treatment response. We actually also trained a separate model on placebo arm, but found that it didn't work at all for the treatment response prediction. So by visualizing the model, regional model weights, so we found a spatial pattern that is actually pretty consistent with our earlier voxel-wise moderation analysis. So together, these results support our hypothesis that fMRI activity during emotional conflict regression can actually serve as a predictive biomarker of antidepressant response, and which offers potential guidance for personalized treatment selection in major depression. So in addition to the task fMRI biomarker study, we also explored whether EEG, which is a more accessible and a cost-effective tool, could also offer predictive biomarkers for antidepressant response. However, EEG-based biomarker study faces several major challenges, including the poor spatial resolution due to the volume conduction effect, and the high spatial temporal dimensionality that increases the risk of model overfitting, and also the fact that feature extraction and modeling are often optimized separately under many existing and conventional EEG analysis approaches, which actually limits the model robustness. So to address this issue, here we develop a new latent space machine learning algorithm tailored to EEG analysis. So essentially, this model jointly learns spatial feature mapping, regression weights under a unified convex optimization framework. So key innovations include the modeling of EEG covariance matrix instead of time series or power spectral features, and also the incorporation of a nuclear norm penalty here. So if we look at this simple linear equation, we can see that if we do the eigenvalue decomposition on the weight matrix W, so the obtained eigenvectors would essentially be the spatial mapping or weighting, and the eigenvalues would serve as a regression weights. Since we imposed, a nuclear norm penalty here, we would enforce a sparsity on the eigenvalues. Essentially, it actually unified the feature mapping, latent feature selection, and the predictive modeling under this one objective. So also this gives us a convex optimization framework to guarantee the global optimal solution. So by integrating all these features, our model directly addresses the core limitations that I just mentioned, and which enhances reliability, interpretability, and clinical potential. So we trained our latent-based EEG model on antidepressant sertraline arm to predict a symptom change. So as we can see, it actually works pretty well for the treatment outcome prediction. And the signature identified was primarily from the eyes open alpha band. And similar to our previous fMRI finding, the EEG signature didn't generalize to the placebo arm, and indicating the treatment actually specific signal. So to visualize the potential for patient stratification, so we divided patients into high versus low predicted response group using a median split. So as we can see for the sertraline group, the high first sertraline on the high group actually reached a significantly higher response rate compared with the low group, but no such difference was observed for the placebo. So to better understand the model, we examined both the scope and the cortical distributions of the two most predictive latent signals. So as we can see, the spatial pattern here were primarily localized to the parietal occipital regions and the lateral prefrontal regions. And so importantly, these findings are actually aligned with previous group level studies that show greater parietal occipital alpha power predicts better SSRI response. So our study here extends this by identifying individual level data-driven EEG biomarkers with direct translational potential for personalized treatment prediction. So next, we're trying to answer two questions. First is whether this fMRI and EEG biomarker findings can be replicated in independent cohorts, and also whether these two type of biomarkers show any intrinsic association with each other. So first, we performed a within modality replication analysis of the EEG biomarker. So we applied our EEG prediction model to a separate data set, including 72 patients. These patients were categorized as either partial responder or treatment resistant. So as we can see that the model actually significantly predicted a higher treatment outcome actually for the partial responder group compared with the treatment resistant group. This actually provided very strong evidence to support the predictive utility of the EEG biomarker. So next, we further tried to evaluate the potential cross-modality convergence. So we examined another data set of 24 MDD patients. The sample size is relatively small, but it offers a very unique resource because it includes both resting EEG and the emotional conflict task fMRI for us to examine this cross-modality question. So we applied our previously trained model to each modality and compared the predicted symptom changes. So very striking, we observed a significant positive correlation between the models from EEG and fMRI. Actually, this suggests a shared antidepressant responsive neurophenotype that is detectable across both modalities. So to further explore the neurobasis of the sertraline predictive EEG phenotype, we examined this relationship with single pulse TMS EEG responses, primarily in the dorsolateral prefrontal cortex, or DLPFC. So here, stimulation sites include anterior bilateral anterior DLPFC, posterior DLPFC, and as well as primary visual cortex and motor cortex as control regions. So we found significant correlations between the EEG phenotype and TMS evoked responses in 3 out of the 4 prefrontal regions, as indicated here. But none of those control regions showed a significant correlation between EEG phenotype and TMS EEG response. So this actually indicates that the EEG phenotype reflects cortical responsivity, specifically in prefrontal areas. So building on this, we further tested whether the EEG phenotype could predict a treatment response to rTMS. So using data from open label clinical study, we found individuals with lower resting EEG predicted improvement from sertraline showed a greater symptom reduction with 1 Hz right DLPFC TMS. So this actually suggests a potential treatment selection strategy. If you look at this EEG predictive symptom improvement as well as the rTMS symptom reduction. So this actually informed that patients less likely to respond to sertraline may benefit more from rTMS treatment, essentially offering a personalized and evidence-based approach to depression treatment. So the 2 studies I just presented primarily focused on either brain activation or power spectral features in specific brain regions. In contrast, brain connectivity, which I believe everyone has been very familiar with, or brain network or connectome offers a complementary way to study brain dysfunction by examining interaction in neurosignals between different brain regions. So this network approach allows us to examine abnormalities in large-scale brain circuits, such as default model, front-right control, and ventral attention networks, and so on. So, and these large-scale brain circuits have been, have been shown to be sensitive to brain abnormalities across different types of mental health conditions. So in the next few slides, I will walk you through some of our recent studies using predictive modeling on brain networks to identify disorder-specific neural circuits. So this work was led by my PhD student, Alex Dow, who is now a postdoc at UCLA. And so this study focuses on individualized brain network modeling using resting state fMRI data from the IMPACT trial. So we tried to examine whether functional connectivity could serve as a biomarker for treatment outcomes in major depression. So to better capture individual-specific brain architecture, we developed a machine learning framework that models individualized functional connectivity. So this is based on the hypothesis that shared brain network patterns across individuals may not be informative for predicting treatment outcome. And however, the individual-specific variation may carry more predictive value. So using an unsupervised group component analysis, we decompose the role connectivity into both shared group level components and individual specific connectivity features. So we expect that predictive modeling based on these individualized features can produce more accurate prediction of treatment response. So let's first look at the common components we decomposed across subjects. So basically, we identified 5 stable components, which show pretty distinct brain circuit patterns. So if we look at the component 1, it was primarily driven by connections, between the default model, ventral attention, as well as dorsal attention network. Component 2 was primarily driven by connections within the visual network. Component 3 mainly involved the connections in the somatomotor network and the middle and lateral temporal cortices of the default model network. Components 4 and 5 are more distributed compared with the first 3 components. So overall, the common features driven by these components were found to significantly correlate with neopersonality, such as neuroticism, extroversion, and openness. So in other words, this type of personality characteristics, although relevant to depression severity, may not provide an informative predictive value for treatment outcome. Because their neural correlates are kind of common or shared across subjects. So by applying the individualized models to the treatment response prediction, specifically for search and placebo arm respectively, we found that the model built on the individualized connectivity actually achieved a significantly better prediction performance compared with the model using raw connectivity. So specifically for the search and predictive neural circuit, we found it mainly involved connections within the default model and between the default model from the parietal control and the somatomotor networks. So the placebo predictive neural circuit involved connections within the default model, dorsal attention, and from the parietal control networks, as well as between network connections involving default model from the parietal control and visual network, as well as somatomotor network. So across both modality, we actually found the right prefrontal lobe serve as a key region across the 2 treatment arms. Additionally, the right insula, supramarginal gyrus and left middle temporal cortex were most critical for predicting assertion response. For placebo arm, the left supratemporal cortex and the cingulate cortices were identified as important for the outcome prediction. And furthermore, we also tested whether combining the 2 neural signatures could guide a treatment selection. So using a cross-arm prediction approach, we estimated both search and the placebo outcomes for each individual and quantified the difference between them. So as we can see, so for each individual, basically we can get the predicted search and outcome and the predicted placebo outcome. And then we can quantify the difference to determine which treatment is the preferred option for this individual. So based on this, we tested whether the subjects who were actually assigned to the preferred treatment arm would show better response rate compared with those subjects who were not assigned to the preferred treatment. So aligned with our hypothesis, as we can see, the subjects who were correctly assigned to the preferred treatment actually indeed shows very significantly higher response rate compared with other subjects. So this further supported our hypothesis that the combined signature could serve as a very good indicator for the treatment selection. So in summary, our study here, by characterizing individualized brain functional connectivity, revealed more powerful biomarkers for predicting responses to antidepressant, sertraline, and placebo. So the combined signature showed a significant promise in guiding treatment selection in major depression. So next, I would like to briefly talk about our recent studies focused on developing, psychotherapy predictive connectivity signature for post-traumatic stress disorder or PTSD. So PTSD is a chronic and disabling condition that can develop after exposure traumatic event, primarily characterized by symptoms such as nightmares, heightened reaction, anxiety, and depressed mood, and so on. So although trauma-focused psychotherapy such as prolonged exposure and cognitive processing therapy remain first-line treatments, their efficacy varies widely across individuals, due to the substantial clinical and neurobiological heterogeneity of PTSD. So development of treatment predictive models by leveraging machine learning on brain connectivity actually can, advance the mechanistic understanding of neural circuits underlying the disease and also its treatment. In particular, doing so with low cost and easy to collect EEG may furthermore, facilitate the clinical translation of such models. So in collaboration with Quorum Veterans Bioscience, here we used the data from a large open label clinical study to test this idea. So in this study, resting state EEG was recorded using pretty high density and 256 channel recording system, but it relies on a salient recording system instead of a gel system, so which provide actually more efficient recording setup. And so the data were collected from patients with PTSD, and we reconstructed the individual brain connectons from EEG channel-space EEG to source-based estimates, and then we further constructed a connecton based on the source estimates, which I will walk you through about the construction details in the next few slides. So here with the source reconstructed connectivity, we built a predictive modeling framework that can jointly perform feature selection and outcome prediction for the psychotherapy treatment response. So, as we applied this model to the different bands of EEG data, we found out the machine learning model identified signals primarily from the theta frequency band, eyes open condition, which produced a significant prediction performance for the psychotherapy outcome. We also performed an inner loop cross-validation to determine what condition actually defined the most predictive signature. So as we can see, the results actually showed very consistent pattern, indicating that the signature was dominated by theta band eyes open conditions, EEG connectivity. And so we further tried to interpret the brain circuit patterns of this signature. So as we can see, this signature was primarily driven by connections involving the default model, ventral attention, and the dorsal attention networks. Interestingly, actually, recent studies using resting state fMRI have also shown that PTSD is associated with functional connectivity abnormalities in these intrinsic brain networks. Also, the theta band EEG were known to correlate with cognitive processing, such as working memory and sequencing. So this indicating that this theta band biomarker can serve as a trait-like signature for predicting individual level treatment response. So to better understand the clinical relevance of EEG signature here, we investigated its association with treatment response and self-reports. So we divided the patients into low and high groups based on the medians of the model predicted outcomes, and examined the strengths of the signature in detecting treatment responders versus non-responders. So as we can see, the high group reached a significantly higher response rate compared with the low group. And also this phenomenon generalized across both proton exposure and cognitive processing therapy. So a further classification analysis confirmed the strength of this signature in terms of detecting responders versus non-responders. And so in addition, we found that those patients with higher predicted treatment outcomes showed a significantly greater pre-minus post-treatment changes in self-reports. So these results actually provide supplemental evidence to confirm the clinical relevance of our EEG connectome signature. So together, all the findings support a path toward clinically translatable and scalable biomarkers that could be used to tailor interventions for individual PTSD patients, and thereby advancing the development of more personalized medicine. So as neurobiological and clinical heterogeneity has been a major barrier to precision psychiatry, so understanding and characterizing the heterogeneity is actually pretty critical to better comprehend the underlying neurobiological mechanisms that drive different psychiatric conditions. So identifying neurophysiological subtypes or biotypes holds significant promise for elucidating the heterogeneity by defining distinct subgroups of patients who may benefit from different treatments based on their shared neurocircuit patterns. So this is a kind of a complementary way, I would say, compared with the previous studies that I just introduced, which primarily focused on building predictive models for individual level treatment response. So here, by identifying subtypes or biotypes, especially across disorders, offers us like a more transdiagnostic way to understand the heterogeneity and discover new and effective treatment options. So here we developed an EEG connectome-based data-driven subtyping framework to identify biotypes in both PTSD and major depression. So this slide shows an overview of our subtyping framework. We first used a filter band strategy to decompose the EEG signals to canonical frequency bands. And to minimize the effect of water conduction, we applied the source localization to convert the channel space EEG to source space signals, and then extracted the regional level EEG connectivity by calculating so-called power envelope connectivity. So as you know, because of the water conduction and source leakage during the source reconstruction, so there might be some spurious correlations observed between vertices, and that might affect the downstream analysis. So to mitigate such like zero lag correlation or spurious correlation, so we applied a subnormalization or strategy to kind of reduce the spurious correlation between vertices which are close to each other in particular, and then calculate the power envelope signal and to construct the power or amplitude correlation to capture the source level connectivity. Since we extracted connectivity from different conditions, including different bands and eyes open, eyes closed paradigms, we essentially have a large number of features to utilize. So to achieve an automatic selection of most informative features, here we apply the sparse k-means clustering to simultaneously select the most important features while doing subtyping identification. So unlike other prior studies that have focused on prior regions of interest or selected features relevant to clinical symptoms, here our study primarily relies on like a data-driven framework and integrating connectivity features from different bands and paradigms. So we first applied our approach to a PTSD discovery cohort. So from the histogram of feature importance, as we can see, the beta band from the eyes open condition provide the most informative features for the subtyping identification. Obviously, other bands contribute to some extent, but the beta band is the most dominant condition. So if we look at if we simply compare the connectivity patterns between all patients and healthy people, we actually observe pretty similar patterns between them. So this actually, to some extent, explain why a traditional classification analysis didn't work well. However, with our subtyping framework, as we can see, we actually identified two pretty distinct subtypes, and one still showed a similar pattern with healthy people, but the other subtype showed pretty distinct connectivity patterns. And this can also be observed from the correlation analysis. So by examining the statistical difference in connectivity patterns between subtypes, we found that they actually were characterized by strong differences in the frontal and posterior regions, and primarily involving the front parietal control, default model, and visual networks. So we next tried to replicate the subtype findings using different cohorts. First, we replicated subtypes using independent PTSD cohorts. As we can see, the connectivity pattern actually is pretty consistent with what we identified in the discovery cohort. So we also tested how these two subtypes would respond to psychotherapy treatment. So as we can see, subtype 1 actually showed a significantly better response to psychotherapy treatment compared with subtype 2. So to further examine the transdiagnostic potential, we applied the same pipeline actually to another cohort, including around 200 MDD patients. So again, we observed a very consistent connectivity pattern compared with what we found in the previous two PTSD cohorts. And so since this MDD cohort involves antidepressant treatment, which allows us to further test how these two subtypes may respond differently to drug medication versus placebo. So we found that subtype 1 responded significantly better to certain medication and then placebo. But for subtype 2, there's no significant difference in terms of the responses to these two treatment arms. So we further conducted a cross-modality validation for these subtypes. So they were identified from EEG. Then we were trying to understand whether fMRI could also show certain subtype-specific pattern. So we trained a classification model based on resting state fMRI connectivity and to classify the two EEG-defined subtypes. So we can see the model actually achieved a pretty decent accuracy, around 85%. And we further compare the connectivity patterns between EEG and fMRI. So as we can see, overall, their patterns are consistent or similar. So in summary, we identified neuroimaging of psychiatric subtypes using a data-driven clustering approach on EEG connectivity. These biomarkers revealed two stable and reproducible subtypes, cutting across traditional diagnosis. And importantly, they also show a difference in terms of their responses to both antidepressant medication and psychotherapy. So essentially can offer a promising and a generalizable signature for, characterizing the neurobiological heterogeneity in these mental health conditions, with potential for translation to, precision psychiatry and clinical implementation. So the next part, I would like to quickly walk you through our study about the brain behavior mapping of psychiatric heterogeneity in ADRD and ASD. So this study is about building the brain and neuropsychiatric symptom association or linked dimension, and further quantify the heterogeneity by identifying biotypes based on these dimensions. So neuropsychiatric symptoms, or NPS, are among the most prevalent features of Alzheimer's disease and related dementia, affecting nearly all individuals across the course of disease. So symptoms such as agitation, apathy, depression, and anxiety contributed to reduced quality of life and higher healthcare costs. Despite their clinical significance, the neurobiological mechanisms underlying MPS remain poorly understand. So here we try to address this challenging by applying a pretty straightforward machine learning framework by linking, brain connectivity and MPS. So we applied like a sparse canonical correlation analysis to identify latent dimensions that link both modalities, followed by performing clustering analysis to identify subtypes. So it's actually a pretty straightforward and simple framework. So we used the data from OS3 and ABD, and included the subjects who have both FMI and at least one MPS at baseline. So we performed both discovery and validation analysis. And as we can see, we identified two cross-validated and generalizable dimensions. So one is primarily driven by behavioral dysregulation subcentual. And if you look at the MPS profiles, so that's how we define these first dimensions. Likewise, the second dimension was primarily driven by the anxiety-related subcentual. So with these two dimensions, we further conducted a clustering analysis to see whether subtypes can be identified based on these two axes. So we identified actually three different subtypes, as we can see here. And then we compared the subtype specific connectivity versus healthy people, and we found actually found the two abnormal subtypes. One is primarily relevant to the behavioral dysregulation. The other is relevant to the anxiety-related abnormalities. So these are the connectivity abnormalities of the two subtypes. For subtype 3, it didn't show any significant difference compared with healthy people. So we actually would like to name it more like a resilient subtype or normative subtype. So we also observed pretty distinct trajectories of these subtypes in terms of their clinical progression. So essentially, the resilient subtype showed more comparable trajectory with healthy people, but the two abnormal subtypes showed like worse trajectory in terms of their clinical decline. So this actually provide like a data-driven approach linking brain and behavioral symptoms to offer us a new approach to better manage Alzheimer's disease patients based on their MPS profiles and potentially advancing the targeted intervention that can specifically target the certain MPS-related brain abnormality to achieve more personalized medicine. So next study is about uncovering brain symptom latent dimension in autism spectrum disorder, which follow a similar pipeline as we used in the last study, but a slight modification is incorporation of the contrastive learning component. So here we tried to disentangle ASD specific dimensions, and so that we expect to be more relevant to clinical symptoms for defining clinically relevant dimensions. So as we can see in this framework, we used ASD patients as a foreground group and healthy control subjects as a background group, and then apply a contrastive PCA that can help us to extract or separate latent dimensions and that are specific to ASD versus those shared with healthy people. So this is the main difference of this approach compared with a standard PCA. So with the disentangled ASD specific latent dimensions, we have further applied a sparse canonical correlation analysis to link those ASD specific dimensions to symptom traits, primarily focused on repetitive behavior, social responsiveness, atypical behavior, communication, and restricted interest, as well as social responsibilities. So this dimensional approach, allows us to identify latent factors that bridge neurophysiological patterns with behavior symptoms, offering like an integrated view of ASD heterogeneity. So in our analysis, we identified two robust brain behavior latent dimensions. One in the EEG theta band. By the way, this study was conducted based on the EEG connectivity, and the other in the alpha band. So the first dimension was primarily associated with the communication and socialization subscales from the Vineland adaptive behavior subscale. And the second dimension was primarily driven by the restricted and repetitive behavior of the adults, with some smaller contributions from the autism impact measure scale. So in other words, the theta band connectivity actually is closely related to social communication deficits, which is a core feature of ASD. On the other hand, the alpha band connectivity actually profile was primarily associated with the restricted repetitive behavior domain. And we further explored the underlying neural circuit profiles associated with these two dimensions. So for the social communication deficits dimension, we found that the most salient connectivity was observed between the left angular gyrus and the right middle temporal gyrus. More broadly, this dimension primarily involved the interactions within and between the default model and the frontal parietal control network. In contrast, the RRB linked dimension showed strongest contributions from the connections between the frontal parietal control, dorsal attention network, and visual networks as well. So in particular, right IPL here emerged as central hub for this circuit. And we found that standard variability across patients primarily reflected subclinical differences, underscoring actually the need for extracting disorder-specific signals using contrastive learning. So contrastive features, as we can see here, showed a significantly stronger association with symptom severity across most clinical measures, indicating their superiority in capturing ASD-related variability. So in summary, our study here offers a clinically meaningful framework for parsing ASD heterogeneity by linking neurophysiological patterns to symptom dimensions. We provide new biomarkers and symptom scores that may support a neuroimaging guided personalized intervention for ASD. So we believe this framework can move us closer to more personalized and mechanism-based treatments for autism spectrum disorder. So lastly, I would like to quickly walk through the study relevant to cocaine use disorder. So CUD is a major global health issue characterized by intensive cravings and impulsive behavior. Despite its prevalence, the neurobiological mechanisms underlying this condition remain, poorly understood. Although prior studies have, explored whole brain connectome-based biomarkers for CUD, none of them have, produced consistently reliable or replicable signatures. So the goal of this study here is to develop a generalizable functional connectivity signature that can accurately detect neural circuit abnormalities associated with CUD and potentially inform treatment response prediction or selection. And so to achieve this, we used recent state fMRI data from a discovery cohort of 129 patient subjects, and apply the XGBoost machine learning algorithm to build a classifier that can distinguish CUD and healthy people. So as we can see, the classifier achieved a pretty decent accuracy with performs above 80%. And by further looking at the connectivity importance, we found that the signature was primarily driven by between network connections compared with within network connections, highlighting for example, from the prior control, default model, and dorsal attention network. So by looking at the connectivity circuit, we found increased connectivity between the front prior control and ventral attention networks, and also between the dorsal attention and visual networks in COD patients compared to healthy people. We also observed decreased connectivity between the default model network and the limbic network in COD patients. We next evaluated the replicability of our classification models on an independent cohort. So as we can see, the model maintained decent performance. The accuracy dropped a little bit, but if we look at the connectivity patterns we identified in the independent cohort, it actually is very consistent with what we identified in the discovery cohort. So to further validate the clinical utility of this signature, we tested its ability to predict a treatment response using data from a randomized clinical trial. So in this trial, 25 COD patients received 5 Hz RTMS to the left DLPFC, while another 20 patients received sham stimulation at the same site. So using pre-treatment resistance data from we trained a relevant spectrum machine-based regression model to predict changes in craving levels as measured by the visual analog scale. So this allowed us to test, whether the identified diagnostic signature could also serve as a prognostic biomarker for RTMS treatment response prediction in COD. So as we can see, the signature actually produced a significant prediction for the active RTMS group, but not the sham group. So as we can see, the prediction is not significant by applying the prediction model to the sham group. So taken together, these findings actually demonstrated that our connectivity-based biomarker is both robust and generalizable, and not only distinguishing COD patients across individual cohorts, but also predicting clinical response to neuromodulation treatment. So this work highlights the potential brain network signatures as clinically actionable tools for both diagnosis and personalized intervention in substance use disorders. So lastly, I would like to thank all my collaborators involved in our study, and also some funding agencies for supporting our work. So with that, I would like to stop here and happy to take any questions. Thank you for your attention.